I then moved to the UK, did my PhD at Manchester, worked at Birkbeck, and worked at Exeter currently. And so I've evolved from looking at minerals in rocks to try and find mineral deposits, to looking at minerals in tailings, which I'm doing at the bottom in Cobalt, Ontario. And over the years, I've loved minerals right from the start, and I've been thinking about and working on minerals for a long time. So I'm going to open up by just reviewing minerals and contaminants. I know I'm speaking to the choir or preaching to the choir, but just a quick review about that. Give you some examples of the research I've participated in and others have done on some of the key points I'd like to raise through my reflections, and then just some future considerations to think about. So let's start out with minerals and contaminants. When I studied geology many years ago, this is what we were taught. We were taught that minerals are naturally occurring, they're inorganic, they're crystalline, and they have fixed or variable compositions. So we've got some lovely quartz there, and we've got the Shap granite up in the Lake District with those beautiful K feldspar crystals. And when I moved to the UK to start my PhD, I heard about this field called environmental mineralogy or environmental minerals. And this was fantastic because it took everything I knew about minerals and added on layers. So the definition of those are they're naturally occurring, or actually they can be anthropogenically derived. They are inorganic, but they can be organically derived or contain organic components. They are crystalline to very poorly crystalline, which makes them interesting and more difficult, I think, to work on. And then, like the true minerals, they have fixed or variable compositions. So a few examples for you there, some banded iron formation, some clay minerals up in the upper right-hand corner with some biofilm or EPS linking them together, a lead-bearing mineral, which I'll talk about later, and some soluble sulfate salts from Cyprus. So a huge range of different types of minerals. And we know that in our environmental mineralogy and in our traditional mineralogy, minerals can sequester contaminants. And we know there's huge sources of contaminants that we're always exposed to. So we've got the picture of the lovely wedding in Mount Isa in Queensland with some lead coming out of the smelter stacks. On the lower left-hand corner, we've got volcanic emissions, so glasses, ash. We've got acid mine drainage in Cyprus on the bottom. We've got urban transport, and we've got deep sea vents. So depending on how you look at this, these elements can be ore deposits, they can be naturally derived or anthropologically derived rather. But if we are exposed to them in excessive amounts, they become contaminants. And because minerals that we know about can take up contaminants, and contaminants can be metals, metalloids, nutrients, anything that's really in excess of what our requirements and ecosystem requirements are, these are called contaminants. And we know that they can be taken up and stored by minerals. And that's why a lot of us are here today. And that's why we study minerals in particular. And because the minerals can take up these contaminants, we know that the cycling of contaminants in our Earth systems is controlled by the stability of minerals. And the stability of minerals is in turn controlled by a number of factors. One that I've got here is bacteria that are sitting on top of an iron oxide mineral. This is from one of Mike Hochella's works, I think. And it's geobacter, it's dissolving that iron oxide. So if that iron oxide contains contaminants, they can be potentially released. But there are lots of other factors that influence mineral stability. There's pH, fluid composition, temperature, and so on. And so we as environmental mineralogists and clay mineralogists, we study these minerals. And that's what we're here for today. So I'm just going to, as I said, go through a number of things that I would like to highlight in my more than 25 years career looking at minerals, and just reflect on those and pick out some of what I think are the highlights. So the first one is, as far as I know, I think everybody has a favorite mineral. Mine is Gyrocite. And some of you heard this before. And Gyrocite, why is it Gyrocite? Well, I started studying Gyrocite when I got my job at Birkbeck, actually, and worked in the Rio Tinto. And there was lots of different types of Gyrocite around. And then I looked into it more, and I found that it's a really cheerful yellow mineral, so that's a great thing. but also in terms of environmental mineralogy, it's a bucket mineral. And that means within its structure, it can take up all types of contaminants. So I've put the structure of gyrocite on there for you. The A site in gyrocite normally contains potassium, but it contains contaminants like lead. The T site normally is iron, but it can contain copper, zinc, chromium, aluminium. And the T site, which is yellow, is normally sulfate in gyrocite, but it contains arsenic as well, or arsenate. So it's a really interesting mineral, and we can study it in all sorts of ways to understand how it sequesters contaminants. But of course, as many of you know, I've got a picture of Mars there as well. In 2004, some Mossbauer spectroscopy on the surface of Mars detected the presence of gyrocite. And there has been lots of studies since to show that gyrocite and other sulfate minerals exist. So it really has an Earth and planetary perspective. So I've been studying gyrocite for a long time, since 2004, when one of my first PhD students, Adrian Smith, who you can see in the upper right-hand corner, started with me and did a PhD in the lab, synthesizing gyrocites and then looking at their stability in different fluids. And so he synthesized pure gyrocite, so that's potassium gyrocite, but also gyrocites that contained contaminants like lead and arsenic. And this is some of his work. So he synthesized the gyrocite before picture that you can see there. It's pretty nice and not a great crystal structure, but it is certainly globs of nice mineral. And then he did some acid dissolution experiments on that. So took the gyrocite out of its stability field into a lower pH. Gyrocite normally forms at low pH anyway, but he took it lower than its stability field. And as you can see, it broke down. It looks like Swiss cheese or something's been chomping on it. And then he tried to look at the products of this dissolution, both the aqueous and the solid products. And you can see there's a surface product on that, and worked out the reaction that the dissolution of this lead arsenic gyrocite at low pH produces a gel surface coating of lead sulfate. So it takes the lead back and makes sure that doesn't get into the water systems, but does release the arsenic into solution. So gyrocite is a temporary store within its stability field of lead and arsenic. Adrian also worked with Kate Wright, who used to be at UCL and is now in Australia, to do some chemical work on the structure, down to the atomic structure of gyrocite. And he did some computational modeling of the gyrocite structure. So you create a gyrocite model in this program at the time called GULP. And then what you do is relax the surface. And this would happen in nature. So the atoms move to their lowest energy state, and that's where they're most stable. But you can see by doing that in the upper right diagram, by conducting that relaxation, you can see that the potassium, which is the blue ion, and the sulfate are at the surface, and the sulfate sticks up. And the surface area there is quite large. It's shown by that band of different color. And that means that that surface is less stable than the surface below, which is the iron octahedral surface. And that means that the dissolution of gyrocite is incongruent. The sulfate and the potassium come off sooner than eventually do the iron oxide. So anything in those sites has the propensity to go into solution much more easily when gyrocite dissolves. We've also been fortunate to work with microbiologists. I know a lot of us do now, and this was some work that we collaborated with people in the University of Windsor to look at the reductive dissolution of the lead arsenic gyrocite that Adrian made. And Christina Smeaton, who was a PhD student at the time, did some experiments on that and showed that there was simultaneous reduction of the iron 3 and arsenic 5 at the surface. The iron 3 reduction was driven by thermodynamics, but the bacteria were responsible mainly for the arsenic 5 reduction via detoxification. So a nice blend of traditional chemistry and microbiology in that instance. So the second reflection I've had over the years is that, again, everyone has a favorite mineral, but I think overall that iron oxides are the champions of contaminant sequestration in most systems, natural systems especially. So here's a photo of a site we work on down here in Cornwall called Wheel Maid. It's a tailings dam that's been abandoned, and it's really close to the campus, so we take students there and we do a lot of work on this site. It's acid mine drainage water that you see in the middle there, and by evaporation and other processes, iron oxides form around the surface or the edge. I've also got a photo from Yellowstone, and yet the volcanic waters at Yellowstone contain all sorts of interesting elements. And again, down the streams, flowing from the vents and the little craters that are formed, you see a lot of these iron oxides. And I've seen this in a lot of the work I've been able to do over the years. This was one of the first projects I had when I got my job at Birkbeck. We went to study the aftermath of the 1998 Athna Kayar tailings dam spill. You can see a picture of the spill on the left-hand side. So this was one of the biggest spills in Europe at the time. And it was pyrite-bearing tailings that spilled from a small mine. The tailings dam failed and spread out all over the river system. So we studied a number of things there, but one of them was to study the speciation of arsenic, what happened when it rained on the floodplain following remediation. So the first arrow points to a little pool, and we sampled the pool, the waters, and the solids. And if you follow the arrows to the picture with the little sunglasses, what we found both on the floodplain was that there were tiny pockets of tailings around, even though they've cleaned off the floodplain to remediate it very well. And those tailings still contain primary arsenopyrite, which you can see in the chemical map. This says arsenic. The really sort of bright blob is arsenopyrite. But ultimately, that arsenopyrite weathered, broke down, and the arsenic was sorbed onto iron oxyhydroxides. So iron was the final product there, and really mostly the final sink of arsenic in that system. Another tailings dam spill that we studied is the 2014 Mount Polley tailings dam spill in Canada. That at the time was the largest tailings dam spill on record, 25 million cubic meters of tailings and water that spilled into this watershed. So you've got a before picture there and an after one at the lower left. And Hazeltine Creek, which you can barely make out sort of in the lower right corner, was a 1 meter wide creek that expanded to 100 meters wide by this volume of tailings moving through. So we were interested in several things. One of them was vanadium, actually, because although these tailings were really not toxic as we classify them, they were pretty, didn't contain many metals or metalloids in high concentrations. They didn't have pyrite, so they weren't acid generating, but they had slightly elevated vanadium. So we were quite interested in that. So we took samples of the tailings, which you can see on the left. They're pretty innocuous looking tailings. And we found that vanadium was hosted by titanite, which is titan in the picture. It's a calcium titanium silicate, and by magnetite. And we also took some samples of the secondary, what we thought were secondary. They were forming from pools flowing underneath the tailings, iron oxide. We found that the vanadium in the iron oxide was vanadium 5, and the vanadium in the titanite and magnetite was vanadium 3. We also did some pore water studies. So this is my colleague Patrick taking some pore lysimeters really into the stream. And just below the tailings water interface, the vanadium was extremely high in concentration, more than 2,000 micrograms per liter. And as a general guideline in waters, the British Columbian guideline was 10 micrograms per liter. So it was fine in the stream, less than 10, but below that sediment water interface was very high. So we put everything together that we had and came up with a model whereby we felt that the magnetite and the titanite underwent reductive dissolution, broke down the minerals to some extent, released vanadium, which then oxidized in the fluid to vanadium 5+, passed through the sediment water interface, and then formed these vanadium-bearing iron oxyhydroxides, my champions. But of course, this is the clay minerals group with us today, and we did have also evidence that there was some vanadium-bearing aluminium hydroxides or possibly clay minerals in that system as well. Another study I've done with, led by Karen Johnson, who's at Durham University, she's an environmental engineer, was to use iron manganese hydroxide waste from water treatment to treat contaminated soils. So some experiments and lysimeters were done on this, and the bottom line is that the waste does very well at sequestering the arsenic. But what happens is the manganese oxide that's in the waste is the thing that oxidizes the arsenic 3, so it's very important. But the final champion in sequestering the arsenic is the iron oxyhydroxide. So you can see there's some chemical maps there on the right-hand side. There is iron and manganese definitely in that waste, but actually the arsenic is sequestered only in the iron oxide. And you can see that by the plot at the bottom as well. So both are important, but we think that the iron oxyhydroxide, and we've shown that by X-axis as well, is really the thing holding the arsenic in the end. But for those of you who like manganese oxides, I think they're the close runners-up in terms of sequestering contaminants. So my PhD thesis long ago, you've seen that photograph on the left, that's a lead-bearing iron manganese oxyhydroxide. And in fact, the really bright areas, which are the lead-rich ones, are actually associated with manganese, not with the lead. And this has been seen by others. So there's a classic study done in Pinell Creek in Arizona by John Barger and colleagues in 2009. This drained a former copper mine, and the manganese oxides, as you can see, form grain coatings. They're largely layered manganese oxides like bernocite, and they sequester the contaminants. So together with Karen again, Johnson at Durham, we've also done some work on manganese oxide water waste and added that to soils containing lead. And you can see in the top chemical map, we've got the manganese oxide that we've added to that soil, and there's no lead before the treatment. So we just add that manganese oxide, and then after 10 months, you can see that the manganese oxide has a lovely layer of lead on the outside. So it sequesters that lead very well. And that's something we see. Lead seems to have a very strong propensity for manganese oxides, or the other way around. How are we doing? Okay. So the other thing to say is, although I'm championing iron and manganese oxides, there are a number of different types of environmental minerals that we know about. And the point of this slide is to say that although we do see that iron and manganese really, as a whole, sequester a lot of the contaminants, there are still many minerals in these systems that are probably just as important in specific cases. And I say many minerals yet to be discovered. These are minerals that aren't new, so they're known about. But when I say discovered in these contaminated environments. So for the first time, we might find a mineral like brandholzite. So the top study I've got there is by Heather Jameson, a colleague at Queen's University in Canada who studies mine waste mineralogy. And she and her student Agnieszka see the stibnite tailings at Beaverbrook in Newfoundland, and actually found that the main mineral controlling antimony mobility was brandholzite, as you can see here. So one that you might not have expected. So it's still worth really characterizing these environments very well. Another example of this is a study by Heather and colleagues and Joanne Santini, and I did a little bit on this one, in Newfoundland, or sorry, not Newfoundland, Northwest Territories. Yellow is after the gold that was found in the Yellowknife area, and a town grew up around the big gold mine in the 1930s. And the gold was sequestered in arsenopyrite, which they roasted to release it. But of course, that released arsenic from the arsenopyrite, which oxidized partially to arsenic 3+, and then was emitted to the environment. So there is evidence of arsenic trioxide contamination in the wider environment, but the actually roasting process produced arsenic trioxide as well. That's stored underground currently in the mine. And Heather went underground and found these lovely biofilms in the right lower picture, which we didn't really recognize as biofilms to begin with until Joanne, who's at UCL, she's a microbiologist, started studying these. And the mineral they found was euconite. And again, I don't know if euconite had ever really been found in these environments, but again, by studying them in detail, they found the mineral and then made a link to the bacteria because Joanne and her student Tom Osborne found this new bacterium GM1, which is an arsenite oxidizer, converting the arsenic trioxide to arsenic pentoxide, which then was taken up by the euconite. So we can see the cycling of arsenic and the importance of euconite in the system. So just to finish off that thought, we can do experimental studies that complement these field studies. This is a study I did with my former postdoc, David Kossoff, and we'd been talking to Dave Craw, who's in New Zealand. And Dave has done an awful lot of work studying mine waste in New Zealand and showed, as you can see in the upper left-hand corner, a really good link between antimony and arsenic in mineralized rocks, in ores, and stream and mine sediments. So they tend to occur together, and they actually occur in the same column of the periodic table, so they do have similar properties. So we did a study on scorodite, which we know is a reasonably insoluble arsenic-bearing mineral, and one that people say is a very good sink for arsenic. And we thought, well, let's see what happens if we put antimony in it and see if we can form something that's equally as insoluble. And interestingly, we didn't manage to get any antimony into the scorodite structure. You can see that we start with pure scorodite at sample 200, and as we progressively add antimony to sample 210, we kept forming a new mineral that looked like an iron oxyhydroxide, actually, just looks like goethite. But in fact, this is tripuiite. And again, tripuiite was known about, certainly, it's a mineral that had been defined. You can see it's an iron antimonate. And the interesting thing about it is it's completely, well, very, very, very insoluble. David tried to digest it in hydrofluoric acid and had no luck. And so as a result of this and many other studies which have seen tripuiite, people are really looking for it now in these systems and seeing that it might be a really good sink for antimony, which of course is very toxic to us. And then finally for the reflections, this one's kind of long. It says processes, I should say, involving minerals and contaminants occur concurrently, or basically everything happens all at once. So this is the Bolivia. This is the Cerro Rico mountain where tin and silver have been mined since 1540 when the Spanish invaded and thought this is a great place to get some silver. So they mined the deposits and they threw the waste into the Rio Pilcomayo, the river Pilcomayo, which you can see in the lower left-hand corner. And that was taken in 1998. And even then, the river ran gray with tailings. Local communities along the river grew crops on these soils. So David, again, who I've mentioned before, did a PhD to study the effects of weathering of these tailings, but also the tailings with soil. And he did a lot of things, but he basically set up the experiments. He was doing it part-time, and he did 20 cycles or 3 years of column experiments, really long time. He'd rain on the columns for 3 weeks and dry out for 3 weeks to simulate wet and dry season. And he found that zinc and cadmium didn't behave similarly, particularly in tailings and soil columns, as you can see. And that was weird. These are similar elements in the same column in the periodic table, so why don't they behave in the same way? So the first thing to think about was the original sulfide. And in fact, he did find that there were 2 forms of ZNS, wurtzite, which had cadmium, and sphalerite, which didn't. So he could have stopped there and thought, well, that's the explanation. It's the weathering of these polymorphs. But the point was that when he really looked at the mineralogy, it was basically everything happening all at once. It is the ZNS polymorph weathering, but it's also secondary iron hydroxides that absorb zinc and cadmium differentially. It's soluble secondary sulfites, and it's also organic acids. So this is a fact about both experimental and natural systems. Everything happens all at once. And we have to try and keep in mind all of these things, but recognize that some things happen faster than others. Okay, I don't have much time, so I'll run through my considerations reasonably quickly. So based on everything that I've been involved with, I think it'd be, and I know these aren't new thoughts, but it would be great to do more mineralogical analysis directly in the field. We have soluble sulfate minerals, we have these white bioprecipitates. If we sample them and take them back to the lab, they can change, just like any other mineral. So going forward, we know there's devices like this HALO device, which my student Richie is using. This is measures iron reflectance, but there is also a spectral library for minerals, and you can do this right in the field. This has to be part of our future. Of course, we would back up things in the lab, but the more we can do in the field, the less disturbed minerals are going to be. And hopefully we can use the technology developed by things on the Mars rover, for example, to really do a lot more in the field. The second thought is that we need more fundamental studies of properties and thermodynamics of minerals. Here's a couple of examples just put up for you. This is bread and butter stuff for us, but it really is important to understand how minerals sequester contaminants, but also to develop the thermodynamic data that we can put into models like Freaksy to predict what happens. Without those data, we can't really get perfect models. So we definitely need to do more of that. I know some of you are talking about organics today, and that's terrific. And there's a lot of work now going on to understand the relationship between minerals that contain contaminants and organics. A lot of this is in the lab for obvious reasons, and some in the field as well. But it came to my mind because I worked on a Chinese tailings that you can see here. This is in Guangxi province in the south of China, where you can see the tailings actually filling up karst valleys. These are enormous tailings dams. And in China, they use a lot more organics to process their minerals from the mines than we do in the West, because the organic are cheaper in China. But as you can see in the cartoon in the lower right-hand corner, this produces a lot of different types of organic compounds in the tailings piles, both initially and as these tailings evolve. And we don't really know the relationship between those organics and the contaminants. And so more of these field studies would be great. And then finally, something I've been really doing a lot at Exeter, which is great because I'm sort of surrounded by people in different disciplines, is to think laterally and to mingle when we have the time and when we can, COVID permitting. So this is John Clark. He's a senior lecturer in creative writing at the University of Exeter. He's a poet mainly. And for some years, he's been working on the Red River, which is a polluted river in Cornwall. And he's got a project to look at poetry in the Red River. So Laura Newsom, who I know is here, and we have been out with John on the Red River in a very wet day. You can see the photograph in the lower right-hand corner. And we had a look at some of the precipitates in the tailings ponds and the ponds that are around the area. And they're we couldn't sample them at the time, but they're probably arsenates. There's a lot of arsenic around. And we're pretty sure that there's some biological activity helping at least to form these precipitates. So we're going out for a walk with John this Saturday, and we're going to sort of listen to his conceptual ideas about the way humans and the way the ecology interacts with these environments. And it's really fun, and it gives you a really good chance to think differently about minerals. Okay, so thanks very much again, and looking forward to hearing all the rest of the talks today. Thanks.
Lovely. Thank you very much, Karen. That was brilliant. I'll give at least one virtual round of applause for that.
Thank you.
So, do we have any questions? Feel free to pop them in the chat. I'll try and keep an eye on that as well. And also feel free to put your virtual hand up as well. I'm just trying to see. Jay, are you able to also scan through to see if anyone puts their hands up? Because there's lots of people obviously in the Zoom, so difficult to see.
Sure, we have. Oh, that's a thumbs up from Kevin. It's not a hand; it's just a thumbs up.
Thanks.
I think there's a question from David. I think.
Oh, and James is also popped in the chat as well. James, do you want to go first, and then we'll go to David.
Sure. Hi, Karen. Nice to talk there. At the end, you were there talking about all these processes happening almost simultaneously in some of these systems. Now, that kind of then makes it feel a bit overwhelming. How do you actually break it down and look at these individual processes and then also wrap them up together? I mean, you could have endless PhDs going on this kind of thing.
You can, indeed.
Your kind of advice there?
Well, I think you have to break them down to begin with. So David, for example, really focused a lot on the sulfides to begin with, and then started looking at some of the sulfides and then the other minerals that were in his tailings, and recognized there were other ones, and then tried to make the links between the sulfides and, say, the iron oxides, for example, when he had coatings on the sulfides. So it's a bit like following the paragenesis of what's happening in your system. And I suppose it's trying to, it sounds really overwhelming, but I guess if you can get a complete set of data, both from fluid chemistry, from minerals, doing some modeling, and then I guess trying to put the pieces of the puzzle together and just trying to decide which of your chemicals play the most important roles in understanding what's happening to the contaminants. it's definitely not easy. And I think we got very lucky with David's project because he had a long time to do it. He did it over, like a part-time student, did it over 7 years. So we had a lot of time to think about that. Yes, we didn't consider the microbes, to be honest. I can see there's a picture coming up. So we didn't actually consider those. So it's incomplete, but yeah, I think, well, the advice is to just try and do as much as you can. If you're doing a PhD, you can't do everything. So I think as long as you, in your methods, state, your limitations and be clear about what you're looking at, then, we know that you can't look at everything. But yeah, if you can, it's really rewarding. So hope that helps.
Great. Jay, did you say someone in the room had a question?
I think David had his hand up, but I think it's down now. So I don't know if David had a question.
Yeah, a very quick question, if I may, Karen. It's lovely talk, lovely to hear about this again. Is much known about the interaction of tailings and seawater?
No, not at all. Yeah, in fact, we are doing a project on it, and I see that Martin's going to talk about seawater and iron, isn't he? We're doing a project about the coastal zone, and actually, you're right. Looking at minerals and interactions with saline waters is really lacking. Really lacking. Yeah.
Thanks.
Great. Okay, we've got a couple of quick questions in the chat as well. So Lucian says, excellent talk. What about the impact of microbes on biominerals formation and dissolution? I see it.
Thanks, Lucian. Yes, I didn't mention it very much, and you're absolutely right. It's completely crucial. I just couldn't quite squeeze it all in. But we certainly need to know a lot more about that. We need to know about what the microbes are, like the minerals. There's, I presume, a lot of microbes we don't even know about yet. And we need to know how they're doing things and what they're doing. So yes, please, equally as important as everything I said, for sure. Yeah.
Right. And then also Mark says, did anyone develop the scarodite process as a commercial route to arsenic fixation?
Oh, Mark, I don't actually know. Yeah, sorry, I don't know if you know. You've got a reference there, 1990s. Yeah, that was I don't know is the answer, but it'd be worth looking at because as long as you don't reductively dissolve scorodite, it's a really good sink for arsenic in soils as well as other processes. So, yeah.
Great. Lovely. Well, thank you so much, Karen.
Okay, thanks again.
That was really
Looking
fantastic.
forward to the talks.
Thanks.